Ah.
Hi there, Professor Volt. Could you go over the requirements for the essay again?
Now we're recording. All right. The cell wall, if it's present, it's usually complex in a prokaryotic cell. And the cell wall, if it's present, it's chemically simple in the cell wall. Like plant cell walls, they're very simple cell walls made of cellulose, and that's about it. I will talk a little bit more of the complexity of the prokaryotic cell wall in a little bit. The plasma membrane, it's present. It's a little simpler, usually no carbohydrates, but it does have proteins in it. And usually it lacks sterols. And then in the eukaryotic cell, the plasma membrane has sterols and carbohydrates. The cytoplasm, this is an oversimplification. The cytoplasm of a prokaryotic cell does have a cytoskeleton. This book is way behind the times. This discovery is now at least 15 years old, maybe 20 years old, where prokaryotes do have a cytoskeleton, but the cytoskeleton is simpler in a prokaryotic cell than a eukaryotic cell. And there's also cytoplasmic streaming in a eukaryotic cell, and I'll show you a video of that. The ribosomes are smaller in a prokaryotic cell. They're larger, ADS in size, in a eukaryotic cell. The chromosome of a prokaryotic cell, usually singular, circular chromosome. The bacteria lack the histone proteins. The DNA of or the chromosome of a eukaryote, usually multiple chromosomes. They're linear, meaning they have a starting position and an ending position. And then the DNA is associated with histone proteins. Cell division, prokaryotic cells mainly divide by binary fission, and eukaryotic cells divide by mitosis. Sexual recombination, there's none in prokaryotic cells, but they can transfer DNA plasmids. And then sexual recombination occurs in meiosis in eukaryotic cells. Any question about any of that? If the cell moves by something that's long, it's a flagellum. If it moves by something that's short, and there's many of them, it's a cilia. Cilia are something you only find on eukaryotic cells, and they're short hair-like structures. They're both used for locomotion. Flagellum can be seen in eukaryotic cells as well as on prokaryotic cells. Eukaryotic cell wall, it is present in plants, algae, and fungi, not in animal cells. It's chemically simple. There are no peptidoglycan molecules in eukaryotic cell walls. We're only going to talk about algae, plants, and some fungi. Cellulose is the molecule which makes up the cell wall. We're not going to talk about the other cell walls. Cell wall is, of course, not present in animal cells. They do have a plasma membrane. The plasma membrane may be covered with a glycocalyx. The cell membrane of eukaryotes is similar to bacteria, meaning the main molecule of the eukaryote cell membrane is a phospholipid. Why is it the same as bacteria? It's thought that a long time ago, the eukaryotes simply used whatever they found and whatever worked well. And they, it's believed, borrowed the cell membrane from bacteria. It's mainly phospholipid. It does have proteins in it. It does show selective permeability. You can have transport across the membrane. The three transports we've already mentioned, simple diffusion, and oxygen and carbon dioxide simply diffuse across the cell membrane. Facilitated diffusion, where a channel protein facilitates the movement of molecules across the cell membrane. You can have osmosis, where water is crossing the cell membrane. You do have active transport, where a transport protein moves a molecule across the cell membrane. And that movement is against the concentration gradient. So the cell has to spend energy. And to move one molecule, it spends one ATP molecule. There is no group translocation in eukaryotic cells. However, eukaryotes can move molecules across the cell membrane by using vesicular transport, where they can form a vesicle and bring in a whole bunch of molecules from outside the cell and inside, or they can have a vesicle fuse with the cell membrane and then dump the particles of the vesicle outside the cell. So you can have vesicular transport in eukaryotic cells. Any question about any of that? There are two types of vesicular transport in eukaryotes. There's endocytosis, where a material moves into the cell by a vesicle. And then there's exocytosis, where a material moves out of the cell via a secretory vesicle. A vesicle brings the molecules up to the plasma membrane and then fuses with it, secreting the molecules outside the cell. Why it's called the secretory vesicle. Okay, any question about any of that? Endocytosis can happen in two different ways. There's phagocytosis, where pseudopods extend around something outside of the cell, engulf the particles, and then bring in the stuff surrounded by the pseudopods, and that'll bring a vesicle inside the cell. And this we can call cellular eating, where the particles are engulfed by a pseudopod, and then a vesicle is made and brought inside the cell to digest. So that is called phagocytosis. There's also pinocytosis, where the membrane folds inward. I don't know if you can see my hands. I'm trying to make the folding of the cell membrane. And a vesicle is made out of that infolding. And then that vesicle is brought into the cell. And that we can call cellular drinking. I thought I had a picture of that, but I apparently don't. All right, any question about endocytosis or exocytosis? If not, let's briefly talk about the cytoplasm. In eukaryotes, the cytoplasm is the aqueous substance inside the plasma membrane, but outside of the nucleus. The cytosol is the fluid portion of the cytoplasm. You can have cytoplasmic streaming, where the cytoplasm moves around in the cell. And this is going to show you a little video of cytoplasmic streaming, where the cytoplasm moves around in the cell.
So I've got my essay written, and I've been working on it for about a week. So now I'm going to show you how I use Grammarly to edit. Okay, so here's my essay, and I'm going to click through.
Sorry, you have to skip the ad. All right, so you can see cytoplasmic streaming, where the cytoplasm is moving around the cell. And it's actually moving chloroplast, which is what we're viewing. So there's chloroplast moving. Can't really make out the central vacuole, but it's most of the cell. Any questions? All right, the cytoskeleton is a structural framework of 3 different types of protein filaments running throughout the cell. It makes it like a backbone of the cell. And in this picture, let me blow this up, you can see that the cell is essentially here. It's only showing the cytoskeleton, but the cell is essentially here. And attached to the cell is the protein backbone of the cytoskeleton. And this one is, we're looking at the F-actin filaments. This one is showing you actin filaments in red, microtubules in green, and the nuclei is blue. So the cell is essentially here. And most of the cell has a cytoskeleton, a network of proteins running through it. Any question about any of that? The cytoskeleton of a eukaryotic cell is very extensive, and much more extensive than you find the cytoskeleton in a prokaryotic cell. Your book may not discuss the cytoskeleton in prokaryotes, but I assure you that it's been known for at least 15 years. The ribosomes are a structure found in eukaryotic and prokaryotic cells. They're a little different in eukaryotes. The eukaryotes have an 80S ribosome, or larger in size. The ribosomes are the site of protein synthesis. In eukaryotes, the ribosomes are either bound to a membrane, like the cell membrane or the nuclear membrane, or the endoplasmic reticulum, or they're free, found in the cytoplasm, sort of floating around in the cytoplasm. If the ribosomes are membrane bound and are a part of the membrane of the endoplasmic reticulum, we call it the rough ER. And why it's the rough ER is that the ER has ribosomes attached to it, and the ribosomes make the ER look rough. If the ER is not rough, we call it smooth ER, and it doesn't have ribosomes attached to it. Any question about any of that? Prokaryotes have a smaller ribosome, 70S in size, and you should note that they are organelles in eukaryotic cells which have ribosomes, and those are the chloroplasts and the mitochondria. These organelles, membrane-bound organelles, have ribosomes in them, and these ribosomes are 70S in size, so that the ribosomes of the chloroplast and mitochondria are smaller than the eukaryote ribosomes. Any question about any of that? When I ever use the word organelle, I mean a membrane-bound organelle. Eukaryotic cells have organelles. This allows compartmentalization of the different functions, and each compartment or membrane-bound organelle tends to have a specific function. They have specific cellular shapes, and they perform specific functions like cellular growth, maintenance, and reproduction. The nucleus, the function is to protect the DNA. The Golgi complex to modify and sort proteins. And we'll talk about each of those in a little bit. All right, let's talk about some of the organelles we find in eukaryotic cells. Most of the organelles are found in both plant cells and animal cells, but there are some, like the chloroplast, which is only found in green plant cells. And there's one, the centrosome, which is only found in an animal cell. It's not found in the plant cell. Nucleus function is to contain and protect the DNA. The endoplasmic reticulum, there's two types, the smooth ER and the rough ER, and they do different things. Rough ER is rough because of the attached ribosomes. The Golgi complex modifies, sort, and packages proteins. The lysosomes is a little vesicle for digesting things. The vacuole is a vesicle that you find in a cell for storing food and other molecules in the cell. It could be waste, for example. In an animal cell, you do find vacuoles. These are small vesicle-like organelles. In a plant cell, and only in plant or plant-like algae cells, you can also have the central vacuole, which is the largest organelle within the plant cell, the central vacuole. It's usually larger than half of the cell, and it's the largest organelle in the cell. For an animal cell, the largest organelle is usually the nucleus. So animal cells do not have a central vacuole, but they do have vacuoles. Plant cells, on the other hand, have a central vacuole, and then they have a vacuole. A vacuole and a vesicle can be the same thing. If the vesicle is moving its contents from one area of the cell to another, we call it a vesicle. However, if the vesicle stops moving and now just stores the molecules at this given location in the cell, we call it a vacuole. So a vacuole is a little cavity for storing molecules, either food molecules or waste molecules. Any question about that? And a vesicle, while it's moving, is a vesicle. But once it stops moving, we call it a vacuole. And now it's just storing things for the cell. Any question about that? The real question is do all vacuoles start as a vesicle? And I don't know the answer to that. But certainly many vesicles switch and become vacuoles. Meaning once they stop moving, we call them a vacuole. Any question about any of that? We'll talk about the mitochondrion and the chloroplast a little later. Perioxisome has specific enzymes for the oxidation of certain organic substances, and the cell can use the perioxisome to destroy things. Like the cell can have hydrogen peroxide in a perioxisome and then dump that hydrogen peroxide against something the cell wants to kill, like an infective agent inside the organism. The centrosome, you find it in animal cells. It's involved in cell division. And the centriole, which is part of the centrosome, actually puts down the mitotic spindle, if you remember what that is in mitosis. Any question about any of this? All right, let's talk about the Golgi complex, the vesicles, and the endoplasmic reticulum. Vesicles start life from usually the endoplasmic reticulum. The endoplasmic reticulum makes lipids or proteins. And if it's the rough ER, it has the ribosomes attached to it, in which case it's making proteins. If it's smooth ER, then it is not making proteins. And smooth ER does not have ribosomes attached. The smooth ER tends to make lipids. Whatever the ER makes, it can be organized in a vesicle, like the rough ER will make the proteins and then include the proteins in a vesicle. And the vesicle just buds off of the ER. And then the membrane-bound organelle called the vesicle transports the proteins or other molecules from one area of the cell to another. And that's what a vesicle does. It moves molecules from one area of the cell to another. If the vesicle contains proteins that were made by the rough ER, these proteins usually end up at the Golgi apparatus. So the proteins are made at the rough ER and taken by a vesicle to the Golgi apparatus. Any question about any of that? The vesicles fuse on one side of the Golgi apparatus. And the Golgi apparatus sort of looks like a stack of pancakes with big globs of butter. Okay? The globs of butter will fuse on this end of the Golgi apparatus, bringing the proteins to the Golgi apparatus. The Golgi apparatus can then sort the proteins and modify them. when the Golgi apparatus is done modifying the protein and sorting them, it will then package the proteins up in a vesicle, which will then come out of the Golgi apparatus from the opposite end of the Golgi apparatus, meaning vesicles fuse into the Golgi on this end, and then vesicles leave the Golgi on this end. So inside these vesicles coming out of the Golgi apparatus will have the finely modified proteins. And then the vesicles will move them from one area of the cell to another. Any question about any of that? Well, where do they go? Depends on the protein. If the protein is needed by the nucleus, the vesicles will fuse with the nuclear membrane, and then the proteins will be dumped into the nucleus. If the proteins are needed by the body, I don't know, let's say it's a hormone or something like that, then the vesicles will fuse with the cell membrane and then dump the proteins external to the cell. And in a teenager who's got active proteins, there will be vesicles fusing with the cell membrane to dump the proteins into the blood that will be outside of the cell. If the proteins are just wanting to be stored by the cell, the vesicles will move to a certain location in the cell, and then they'll stop moving and just start storing the proteins for later use, in which case the vesicle has stopped moving, and now we're calling it a vacuole, storing the proteins for later use. Any question about any of this? So you can see the endoplasmic reticulum, the vesicles, and the Golgi apparatus are all connected together. And they involve the making of the protein, the transport of the protein, the modifying of the protein, and then the final transport of that protein. Any question about any of that? So in eukaryotic cells, we have an endomembrane system. And this involves the transport of different molecules by the cell, especially proteins. Any question about any of that? All right, this is showing you the endomembrane system where a protein gets made by the rough ER. Protein gets made into a transport vesicle, which fuses into this end of the Golgi apparatus. The proteins are then brought into the Golgi apparatus and they're modified. And then the proteins are packaged in a vesicle, which buds out of the Golgi apparatus on this end. And then the vesicles will move the protein to where they're needed. In this case, it's showing you a secretory vesicle. This vesicle will fuse with the cell membrane and then dump the contents of the vesicle external to the cell. Any question about any of that? If not, we've already talked about vacuole. A lysozyme looks similar to a vesicle or a vacuole, but it's more specific in its purpose. And the lysozyme stores enzymes for digesting things. One of the enzymes you find in a lysosome is the enzyme lysozyme. And that's where the lysosome gets its name, from the enzyme lysozyme. All right, here we're looking at the mitochondrion. This is a special organelle, and it's special because it has two membranes, an outer membrane shown here, and then an inner membrane showing you the inside of the mitochondria. The mitochondrion is the site of cellular respiration. It's where the majority of the cell makes its ATP. So it's the cellular powerhouse of the cell, of eukaryotic cells. Here we're looking at the electron microscopic image of the mitochondrion. And you can see that the inner membrane is highly folded, showing you the cristae on the electron microscopic image. The cristae is the folding pattern of the inner mitochondria. The mitochondria is different in that it has two membranes, an inner and outer one. It also is different in that it's an organelle that contains both DNA and 70S ribosomes. Excuse me. There's only one other organelle in eukaryotic cell that is this way, and that's the chloroplast that has two membranes, an outer membrane, and then an folded inner membrane that we call the thylakoid disc, highly folded inner membrane. Inside the thylakoid disc, you can find the granum, composed of one stack of the inner membrane of the chloroplast, meaning the inner membrane is folded a lot in the chloroplast. Inside of the chloroplast, as well as inside of the mitochondria, you can find DNA and 70S ribosomes. Any question about any of that? All other organelles do not have DNA or 70S ribosomes. All right, let's talk about how the eukaryotic cell obtained its mitochondria. A long time ago, I should probably do the artist way. Close that. Let's go. There we go. A long time ago, we had a proto eukaryotic cell, meaning it was a very big cell. And it wasn't a eukaryote, at least not yet. So we're going to call it a proto, meaning before eukaryote. And this proto-eukaryotic cell saw a little cell. I'll just make it here. And it engulfed it to probably eat it later. And it engulfed it with phagosomes so that the little bacteria cell has its own cell membrane as well as a vesicle membrane, meaning it ate it by pseudopods and then brought it into the cell. Any question about any of that? Well, the bacteria, let me put that here. The bacteria could perform aerobic respiration the proto-eukaryote could not. How it made its energy by fermentation. So the aerobic respire, meaning the bacteria, could generate 36 to 38 ATP per molecule of glucose. The proto-eukaryote could only make 2 molecules of ATP per molecule of glucose. The proto-eukaryote had glucose in it. And some of that glucose was taken up by the bacteria cell. And then the bacteria made 36 to 38 ATP. Some of that ATP was released by the bacteria and then went into the cytoplasm of the proto-eukaryote. Now the proto-eukaryote said, wow, I lost 1 glucose, but I made a whole bunch of ATP. We don't know how much ATP the bacteria released, but it was someplace between 2 ATP and 36 or 38 ATP. The point is the bacteria released a lot of ATP, and the proto-eukaryote only lost 1 glucose. It said, geez, I lost 1 glucose, I can only get 2 ATP out of 1 glucose. And this bacteria took up the glucose and then released a whole lot of ATP for me. I'm going to keep this bacteria around because it's making me ATP more than I can make. And so the two, the proto-eukaryote and the bacteria, started living in symbiosis, meaning they started living together. Now, they've lived together for so long that we don't see a proto-eukaryote without the bacteria. So we can finally change the name. It's now become a eukaryote. Let's make a little arrow here. And conversely, we don't ever find the bacteria free living anymore. We only find this bacteria inside the eukaryote. This is how we believe that a eukaryote cell obtained its mitochondria. A long time ago, it engulfed a bacteria that could engage in aerobic respiration. It made the proto-eukaryote more ATP than the proto-eukaryote could make from glucose. And so the two started living together. And now we don't find the bacteria to be free living. We only find it to be the mitochondria inside of a eukaryotic cell. And we don't find the proto-eukaryote to be without the bacteria. It always has the mitochondrion within it now. Meaning symbiosis, they live together for so long that they can no longer live apart, or they at least no longer choose to live apart. Any question about any of that? That is the theory for how organelles like the mitochondria became a part of a eukaryotic cell. And this is known as the endosymbiotic theory. Is there any evidence for this theory? Well, yes, there is. First, when we take a look at the mitochondria, it has two membranes, an inner membrane and an outer membrane. Come back to my thing here, which is exactly what we'd expect if an organelle was brought into the cell by a phagosome, that there would be an outer membrane as well as an inner membrane. There's some other evidence too, and that is there is one eukaryote that does not have mitochondria. What that eukaryotic cell does, I think it's an amoeba, is it engulfs a smaller prokaryotic cell like a bacterium, and then uses that bacterium to make ATP for the cell. And we've seen this before where a protist can incorporate a bacterium and then use the bacterium to do things for the protist. There's some other evidence too. The growth of eukaryotic cells is by mitosis, but the growth of the binary prokaryotic cell is by binary fission. The growth of the mitochondria and the chloroplast are by binary fission. The ribosome of a eukaryotic cell is 80S. The ribosome of a prokaryotic cell is 70S. The ribosome of a mitochondria and chloroplast is 70S. In a eukaryotic cell, the first amino acid made in a protein is the amino acid methionine. In a prokaryotic cell, the first amino acid made is formylmethionine. It's a similar but a different amino acid. In the mitochondria and the chloroplast, the first protein made in the mitochondria of the chloroplast is the amino acid formylmethionine, sorry. In a eukaryotic cell, the DNA is surrounded by histone proteins. In the bacteria, there are no histone proteins in the DNA. There are no histone proteins in the DNA of the mitochondria and the chloroplast. And then saving the last for least, because it's the most, the eukaryote, when we look at the DNA, it's linear. And when we sequence the DNA, it falls within the eukaryotes. When we sequence the DNA of a bacteria, it's one circular molecule of DNA, and it falls within the domain bacteria. meaning the sequence of the DNA. When we sequence the DNA of a mitochondria or a chloroplast, it falls within the domain bacteria, meaning the sequence of the DNA in the mitochondria and the chloroplast is similar to, and most similar to, the DNA of another bacteria cell. And then that DNA is circular. Any questions about any of that? All right, so that's the endosymbiotic theory for how an animal cell obtained an aerobic bacteria, and the two lived in symbiosis for so long, we don't find the mitochondria outside of the eukaryotic cell anymore. And essentially, eukaryotic cells do not have any, are not missing a mitochondria with the exception of one species. They all have mitochondria living within them. And then for plant cells, this evolution of an organelle being taken up by the plant cell actually happened twice, because the mitochondria is in all plant cells, and then the chloroplast is in only the photosynthetic green plant cells. So that the plant cell took up two bacteria, one that went on to become the mitochondria, and the other which went on to become the chloroplast. Any question about any of that? If not, that's it for chapter 4. I think we'll stop a little early today, and we will start at 6:30 with the lab. All right, if there's no questions, I'm going to say goodbye.